Hello, everyone, and welcome to our talk today. But before I begin, let me tell you a little story about Bob and Doug. Now, Bob and Doug have been best friends since they were children and have grown up in the same neighborhood together. They are currently in their 40s and believe they live a pretty healthy lifestyle. They eat right, they have no history of smoking, and they also enjoy exercising on a regular basis. Now, one of the activities that they enjoy doing together is taking a weekly jog around the neighborhood. However, one day, while Bob and Doug were taking their weekly jog, Doug was having some difficulty keeping up with his buddy Bob. Now, this was very disconcerting to Bob and Doug at the time, but they didn't think much of it, and they just continued on with their usual routine. However, in a few days' time, Doug became quite sick. He started having a very irritating cough, some shortness of breath, and also some mild chest pains. Now, Doug didn't think much of this at first and just attributed it to the fact that he might have had a stressful week at work and that he might be under the weather with a cold or a flu. However, as the weeks progressed, Doug's condition worsened. One day, when Doug was getting ready for work, he spat up blood in the bathroom sink. Now, this raised some alarm bells in Doug's mind and led him to seek medical attention from his doctor. His doctor then conducted a series of diagnostic tests to, unfortunately, determine the worst-case scenario possible. Our relatively healthy and young man, 40-year-old young man, was diagnosed with a rare form of lung cancer. Now, you might all be wondering, how did Doug get diagnosed with this disease? And what are his treatment options from this point forward? So we at iPara Innovations are going to talk to you today about increasing adoption of Xalkori, which is an anti-lung cancer personalized therapeutic that will be explored as a potential treatment option for Doug's rare form of lung cancer. The specific objective of our talk is to increase sales of Xalkori, which is, as I mentioned, a targeted lung cancer therapeutic within Canada. And we wish to do this by implementing a pilot study that has a timeline of 2 years, a budget of approximately $200,000, and a positive net present value. Now, we will begin the talk today by highlighting some background information about lung cancer and the various subtypes of this disease. We will then move into exploring Xalkori, or crizotinib, a little bit more, and look specifically into how it targets ALK-positive non-small cell lung cancer. We will then move into the diagnostics aspect that accompanies this drug, specifically the fluorescence in situ hybridization diagnostic test and the immunohistochemistry screening test. We will then move into proposing our business plan, which involves increasing market access within Ontario of this particular drug. Now, let me give you a statistical picture of what is happening in Canada in terms of lung cancer. Now, lung cancer is the most commonly diagnosed cancer in Canada after non-melanoma skin cancers, and it accounts for 27% of all cancer-related deaths. As you can see, 20,000 Canadians die each year from lung cancer. It is also worth noting that the estimated total incidence of lung cancer for 2014 is pegged at 26,000 people. So this just really highlights the seriousness of this disease in Canada and the fact that it needs to be addressed very urgently. Now, lung cancer can be looked at from two different perspectives. We could look at it from a histological perspective and from a genetic perspective. So let me take you through the lung cancer from a histological classification point of view. Lung cancer can be divided into two types. We have small cell lung cancer, which accounts for 10 to 15% of lung cancers. And this type of cancer affects the central region of the lung, and specifically the bronchi region. We also have the non-small cell lung cancer category, which is the majority of lung cancers, accounting for 85 to 90%. Now, as I mentioned, Xalkori is a drug that targets non-small cell lung cancer, so for the purposes of our talk, we will be focusing specifically on this subtype of lung cancer. NSCLC can be divided into three types. We have squamous cell carcinoma, which accounts for 25 to 30% of NSCLCs and affects the thin squamous cells that line the lungs. Some identifying features of squamous carcinoma include coughing up blood and is also tend to be found in individuals with a history of smoking. We then have large cell carcinoma, which occurs in 10 to 15% of NSCLC patients, and this is the least common type of NSCLC, and it tends to affect various regions of the lung and doesn't have a specific region that it targets. Finally, we move into the most common type of NSCLC, which is adenocarcinoma, and this is 40% of non-small cell lung cancers. And this form of carcinoma specifically affects the cells that line the alveoli of the lungs. And as you may know, alveoli are very critical structures to the lung that are involved in gas exchange and so are critical for our ability to breathe. And then we have the alveoli are also important because they have cells that produce mucus. And now mucus is an important substance because it coats these alveoli cells so as not to allow them to become damaged. And this is definitely important in helping us to breathe normally. So now that I've looked into the histological perspective of lung cancer, I will look now further into the genetic classifications of lung cancer, specifically from an NSCLC perspective. So non-small cell lung cancer and certain subtypes of it specifically can be classified by certain genetic mutations. I'd like to draw your attention towards the KRAS mutation, which accounts for about 40% of NSCLCs. We then have the EGFR mutation, which accounts for about 13% of NSCLCs. And as I mentioned earlier, Xalkori targets a ALK-positive non-small cell lung cancer. So for the focuses of our talk, we will particularly focus in on the ALK gene, which accounts for 5% of non-small cell lung cancers. Now, before we look further into the details of ALK, we would like to take you through the treatment algorithm and the molecular testing that occurs to diagnose an individual with ALK-positive non-small cell lung cancer.
So before I get into the intricacies of the treatment algorithm, I'd like to direct your attention to anything that is presented in blue during the pathway will represent Doug's specific treatment algorithm, and anything in gold or yellow will represent the treatment algorithm that can be followed by other patients that may present with lung cancer. So when Doug presented himself at the hospital and his physicians ordered a CAT scan, the CAT scan revealed that he had a peripheral mass. Once this was determined, a needle biopsy procedure was ordered, and this procedure follows a lung aspiration methodology where the tissue is extracted. In the case of other patients that identify with a central mass, a bronchoscopy procedure is followed in order to extract enough tissue. So now that Doug's tissue has been extracted, it was sent to the pathologist and cytologist in order to determine if the mass was truly a cancerous tumor. At this point, it was revealed that Doug actually had tested positive for a cancerous tumor. He was immediately then put on chemotherapy, and this is specifically first-line platinum-based chemotherapy. In the meantime, as his treatment progressed, he was ordered a thoracic surgery in order to extract tissue for a secondary histological classification. In his case, he was identified to have an adenocarcinoma. And as we saw previously, an adenocarcinoma is specified and falls under the category of non-small cell lung cancer patients. So when any patient is identified as a non-small cell cancer patient, they're then subjected to molecular testing. The molecular testing is performed in order to determine if there are any particular biomarkers present in the mass that could potentially have a more targeted and efficient second-line treatment. So in the case of lung cancers, there are two specific types of biomarker testing that are performed. So in Doug's case, he was subjected to ALK testing as well as EGFR testing. And these are performed simultaneously in order to reduce the turnover time for the results. it was revealed that Doug had a negative EGFR mutation. However, unfortunately, he tested positive for an ALK mutation through the screening process, which is called the immunohistochemistry. These results then need to be further confirmed using the gold standard FISH diagnostic assay. And these revealed that he was indeed positive for the ALK mutation. At this point, his physicians began to notice that he was starting to progress off of his first-line therapy. And since he had been proven positive for an ALK mutation, that he could then be considered for a second-line therapy, a more targeted therapy that we know as Xalkori. So just to summarize the treatment algorithm a little bit, when an individual presents themselves with a mass, they undergo biopsy in order to extract tissue for histopathological testing and analysis. Once it's determined that they have a cancerous tumor, they're then subjected to chemotherapy, and their tissue is further analyzed to determine if they're non-small cell lung cancer patients. And those that prove that they are will then go through molecular testing for ALK, and once ALK is found, once they're found to be positive for the ALK mutation, they're then given the Xalkori as a second-line treatment once they've progressed off of chemo. So to understand particularly why Doug developed this particular tumor, we need to look at the ALK protein itself. So first we'll begin by looking at the normal ALK protein, which is known as anaplastic lymphoma kinase. This particular protein is generally active during the early developmental stages of an individual. This protein has 3 specific domains, and the purple domain represents the extracellular domain, the blue represents the plasma membrane spanning domain, and the green is the catalytic domain, which in this case is specifically the tyrosine kinase domain. So for this protein to be activated, it requires a ligand binding to the extracellular domain, which activates another ALK protein, causing the dimerization. This dimerization is stabilized, causing autophosphorylation of the tyrosine kinase domain, leading to down signaling pathway activation. Among this down signaling pathway, there's 2 specific ones, are PLC gamma and RAS, which lead to cell proliferation. And then we have the PI3 kinase, which leads to cell survival. So to further look at what happened and what's wrong with Doug in particular, we'll have to look back at his genetics and the particular mutation that occurs. So in the case of Doug, his ALK gene, which is located on chromosome 2p23, gets translocated and fuses with an EML4 promoter region, causing a fusion protein to be expressed. So now this fusion protein is very different from the one that we previously saw. This one no longer has an extracellular domain. Instead, it has an EML4 domain at the very top, and at the bottom in the green represents the tyrosine kinase domain of the ALK protein. So this fusion protein causes constitutive activation of the ALK protein, leading to extensive cell proliferation. So to look at that in more detail, I'd just like to go into the molecular biochemical pathway of this. So here, as we recall back, the normal ALK protein required that a ligand bind to the extracellular domain in order for it to phosphorylate and then lead to cell survival. However, in Doug's particular case, his protein is located in the cytoplasm specifically, and the fusion characteristics of this protein causes increased stability within the protein, and therefore it no longer requires a ligand binding to the extracellular domain in order to be activated. So this leads to extensive activation of the protein, leading to increased cell survival and increased cell proliferation. So as we can see, this is definitely what led to Doug's tumor development. So when the industry discovered this particular pathway, they looked at particular inhibitors to target this potential phosphorylation of the fusion protein. So researchers developed a molecule which is now known on the market as Xalkori. This molecule is a small tyrosine kinase inhibitor, and in simplified version, it actually targets the phosphorylation, preventing the fusion protein from phosphorylating, therefore causing cell cycle arrest and preventing any further cell proliferation.
Now, how did Pfizer determine that this specific molecular pathway could act as an effective target for specific malignancies? Moreover, how could they determine that the ALK positive mutation would lead to non-small cell lung cancers and act as a biomarker? Well, Pfizer decided to do a phase 1 clinical trial on the effectiveness of this in various tumors. Specifically, they looked at the c-Met and ALK and ROS pathways. Now, for our purposes, we'll define a biomarker as a genetic variant that is associated with a particular disease state. A predictive biomarker being any genetic variant that is going to be predictive and allow us to identify subpopulations of patients that will more likely respond to a given therapy. So looking at this, they tested it along the 3 different pathways and enrolled 550 different patients. Now this is different than a normal phase 1 clinical trial where healthy individuals are recruited. This recruited 550 patients with a heterogeneous mixture of histologically proven malignancies. Measuring the outcomes is adverse events, such as adverse events, serious adverse events, and treatment-related adverse events, as well as serum concentrations of the drug. This allows them to get a safety profile as well as a pharmacokinetic profile of the drug. This confirmed that the ALK is a predictive biomarker for non-small cell lung cancer. Furthermore, they analyzed it in a phase 3 clinical trial in order to prove the clinical efficacy of this on non-small cell lung cancer patients. Now first, they recruited 347 patients who had all been shown to progress off of their first-line platinum-based chemotherapy. Next, these patients were defined to have the ALK positive mutation using the fluorescence in situ hybridization diagnostic assay that we had talked about previously. Now they know that they had to receive second-line therapy and positive for the ALK mutation. These 347 patients were then randomized between 2 groups, either chemotherapy or Xalkori, with the chemotherapy group being broken up into 2 separate arms based on whether or not they had received pemetrexed previously. So they were basically split up into pemetrexed or docetaxel. Each treatment was then rotated on a 3-week cycle, giving us the 108-week timeline that we can see on the left, as the study was conducted between February of 2010 and March of 2012. Now, in order to continue on the accelerated timeline that this drug had for approval, they had to pick a specific endpoint that would allow them to be FDA approved with it, and this is progression-free survival as measured by response evaluation criteria in solid tumors. And this is an FDA-approved surrogate endpoint for phase 3 clinical trials. Now let's look at the progression-free survival. Now, if you look at the y-axis, you can see the proportion of patients that progress off of their therapy. And on the x-axis, you can see the time to progression in months. Now let's look at chemotherapy, which is outlined in red. You can see immediately that there's a sharp decline in the amount of patients that are still on their treatment as they progress off of this treatment. In blue, we can see Xalkori, also known as crizotinib, which shows an increased progression-free survival. So for our friend Doug, if he was put on Xalkori, he would increase from 3 months to 7.7 months, which was shown to be statistically significant to less than 0.001. Now, an increasing trend in pharmaceuticals is not only proving clinical efficacy, but also safety data for the drug, which is what they did next. They recorded patient-reported outcomes in order to determine the safety of the drug. And if we look at the y-axis, we can see the mean change from baseline. Now, a positive number in symptoms represents a decrease in patient benefit, with a negative number representing a decrease in symptoms. However, if we get to global quality of life, a positive number represents an increase in patient benefit, with a decreased number representing a decrease in patient benefit. And I would like to draw your attention to the symptoms on the x-axis, specifically dyspnea, coughing, and fatigue, which Doug showed on his initial run with his buddy Bob. And as you can see, in crizotinib, these 3 symptoms have been shown to have a decreased symptom outcome, showing that after this treatment, he can hang out with his buddy Bob a bit more and maybe get back to his jogging life like he did. Specifically, I'd like to focus your attention to the global quality of life for these patients. You can see here that crizotinib has an increased global quality of life, while chemotherapy has a decrease, showing that not only is this drug clinically effective, but also increases the global quality of life for patients. Based on this data, this drug was approved along an accelerated timeline, indicated as a monotherapy for use in patients with ALK-positive, locally advanced or metastatic non-small cell lung cancer. And specifically, it was approved alongside its companion diagnostic that was used in the phase 3 clinical trials, which was the fluorescence in situ hybridization assay. Now that it had been approved by FDA and Health Canada, could it be reimbursed? This drug was reimbursed as a second-line therapy by the Ontario government with the annual cost of $107,000, as well as the cost of its companion diagnostic of $400 also being reimbursed. So now that we've given you some background on the lung cancer and its subtypes, as well as the anti-cancer therapeutic drug itself, Xalkori, we'd like to highlight your attention to the fluorescence in situ hybridization assay that is used to diagnose these patients, as well as the immunohistochemistry assay that is used to screen for them.
Now, I would like to talk about how fluorescence in situ hybridization, or FISH, is used to detect ALK rearrangement. But before I do that, I would like to give a brief overview of what FISH is and how it works. FISH is an assay that involves the use of DNA probes to identify gene amplification or translocation patterns. In the diagram above, if you look at the bottom of the image, you can see a double-stranded DNA probe. Now, this probe is complementary to the gene of interest, which would be ALK in our case. And the probe is also tagged with a fluorescent tag, which is illustrated by pink color. Now, if you look at the top of the image, you can see the chromosome, which has the gene of interest. And the double-stranded DNA on the chromosome, as well as the double-stranded DNA on the probe, are both denatured into single strands. And as you can see in the middle of the image, the single-stranded probe DNA is bound to the single-stranded chromosomal DNA in a complementary fashion. And once bound to the chromosomal DNA, the probe, which also has the fluorescent tag on it, can be visualized under a confocal microscope, as you can see at the bottom right of the image. And the location of the gene of interest can be determined. Now let's look at how FISH is used to determine ALK rearrangement, which results in EML4 ALK fusion protein. First, let's focus on the image on the top right. This is the full-length ALK gene. The red color depicts the 3' region of the gene to which the red probe will bind, and the green represents the 5' region of the gene to which the green probe will bind. And the gray area in the middle is the ALK breakpoint region. So when a rearrangement takes place, ALK gene breaks in this gray region. Now let's visualize that by looking at the chromosomes on the left-hand side. Let's focus on the first chromosome. As you can see, again, the red and green colors represent the ALK gene. Blue is the EML4 gene. Now when rearrangement happens, ALK breaks in the middle, and the green part of ALK translocates and fuses with the EML4 at the bottom, which is shown in the second chromosome. Now, if you compare the two chromosomes, in the first one, ALK is intact, so you have the red and green colors adjacent to each other, whereas in the second image, because of the rearrangement and translocation, the green region moved further down, and now the red and green regions are further apart from each other. And that's what you can see in a real FISH assay image. So these round cloud images are individual cells, and the red and green colors represent the ALK-specific DNA probes. If there's a rearrangement or split, as is shown with the top arrow, you can see that the red and green colors are apart from each other. However, if you look at the bottom arrow, which shows a non-split scenario, you can see that the red and green colors are close to each other, and they look as partial yellow under the confocal microscope. So in conclusion, the top arrow represents an ALK rearrangement scenario, and the bottom arrow represents a wild-type ALK. Now, FISH is a gold standard for ALK testing. However, it is a relatively expensive assay. It costs about $400 per testing. So in Canada, immunohistochemistry is also being used to test ALK mutations. So basically, NSCLC patients would first be tested by immunohistochemistry, and those patients who test positive in immunohistochemistry would then be tested by FISH to confirm the presence of ALK mutation. So then let's also look at what immunohistochemistry is and how it works. Immunohistochemistry, or IHC, is an assay that used to detect protein expression levels. And here's a brief overview of how it works. Basically, a paraffin-embedded tissue would be deparaffinized to expose the antigens. The sample would then be treated by primary antibodies, which would be specific to the protein of interest, which would be ALK in our case. And then the sample is then treated by a secondary antibody, which is specific to the primary antibody. And this secondary antibody also has an enzyme conjugated to it. And when you add a substrate that is specific to this enzyme, the enzyme will catalyze the substrate into a colored product. So if your protein of expression is expressed, you can visualize the color under the microscope, which you can see here in this next slide. These are ALK immunohistochemistry images from one of the studies. Now, because ALK, or actually proteins in general, can be expressed at different levels, there is a standardized IHC scoring system that assigns a score based on the protein expression level. And the scale is from 0 to 3 plus. So image A has a score of 0, which means there is no ALK expression. And if you go from image B to image D, you can see increasing color intensity, which means increasing ALK expression pattern. So there's no expression of ALK in image A. There's slight expression in B, higher expression in C, and the highest expression of ALK is observed in image B, which would be the case for EML4 ALK fusion mutation. Now that we covered how FISH and IHC are used for ALK testing, let's look at how they are being implemented in Canada, which is illustrated by this graph here. The x-axis shows the timeline in this particular study. The y-axis on the left shows the frequency of IHC testing, which is illustrated by the blue line. The y-axis on the right corresponds to the frequency of FISH testing, which is shown by the red line. And the green line at the bottom represents the number of patients with ALK mutation. And the light blue line, also at the bottom, shows the number of patients prescribed with Xalkori. So adoption of IHC as an initial screening method started in May 2012 and has increased since its implementation. And as IHC adoption increased, there was also an increase in the capture of ALK mutant patients, which in turn translated into higher Xalkori prescription. As you can see, the green and light blue lines at the bottom overlap, which means almost all ALK patients were prescribed with Xalkori. So that's a good trend, and that's good news. And now the question is, what else can be done to further increase the adoption of Xalkori?
So as Emin alluded to, there's been a staggering increase in the amount of IHC screening that is done for ALK-positive non-small cell lung cancer patients. In addition to this, the FISH assay has been used to confirm these patients and has shown a steady increase in the prescription and therefore treatment with Xalkori. So then what's the issue that Pfizer is facing? As a team, iPara Innovations decided to focus on the province of Ontario in order to address and optimize the amount of sales that are being done. This allowed us to look at the geographical distribution of healthcare authorities in order to stratify the population based on not only location, but accessibility to this drug. Just to reiterate, our goal is to increase the sales of Xalkori for the treatment of ALK-positive non-small cell lung cancer patients. And through our research, we found that there was an underserved region in the Northern Ontario area, and due to the stratification of the population, we could narrow down based on local health integration networks, which are regional authorities that are in charge of not only the distribution and funding, but also the integration of healthcare networks. And specifically, Ontario is divided into 4 local health integration networks, or LINs. As we can see here, This is the 14 LIN map for Ontario, and specifically, we're focusing on the northern population, allowing us to focus on the northwestern one, or the northeastern one, sorry. We compared the northwest to the northeast and determined that the northeast would be the most viable for this project as it had a higher population, allowing us to more significantly increase the sales of Xalkori. We then wanted to compare this to the Toronto Central LIN, as this was the gold standard for Ontario at the moment, as it has the highest availability of resources, both human and laboratory. So let's look at that further. We can look at the Northeast LIN as having a population of 565,000, with this being more than doubled in the Toronto Central LIN. On top of this, the Toronto population has access to 5 different diagnostic programs, whereas the Northeast one only has 1, Health Sciences North and Sudbury. Therefore, the population per diagnostic program is actually lower in Toronto than in the Northeast. Furthermore, the testing in the Toronto Central LIN has been known to be every 3 to 5 days for the ALK positive mutation, whereas in the Northeast it is very infrequent and only once a month. Furthermore, based on the geographical location, there's a high access to this testing frequency in the Toronto area, whereas based on both the location as well as the testing frequency, there's low access. So now that we've highlighted where the gap is, we'd like to tell you where this fits into the treatment algorithm that we've highlighted previously.
So when Doug goes to his physician to seek medical attention, his physician would determine that he had a peripheral mass, and as a result order a biopsy sample to be taken. Now this biopsy sample would then be subjected to histopathological analysis, and this would determine that he was indeed an NSCLC patient. Now once he's determined as an NSCLC patient, he would automatically be put on chemotherapy as a first-line therapy, as we alluded to earlier. However, it is in the next stages of the treatment algorithm that we determined that there exists a critical gap within the Northeastern LIN. Now, in the Toronto Central LIN, all NSCLC patients are reflex tested for other biomarkers, such as the ALK rearrangement. And as a result, they'd be prescribed Xalkori if they tested positive. So let's break this down more quantitatively, looking at the Toronto Central LIN as a control. Based on their population of 1.1 million, Cancer Care Ontario projects that approximately 1,200 lung cancer patients would be detected in a year. Factoring in the 85% NSCLC incidence rate, this brings us to a total of 1,025 potential NSCLC patients. Now, assuming that 65% of these patients who would be put on first-line platinum-based chemotherapy would progress, and factoring in the 5% ALK incidence rate, this gives us a total of 33 ALK-positive patients. Now, as I mentioned, since reflex testing is done for biomarkers, such as ALK in the Toronto Central LIN, this gives us a 100% capture rate. However, if we look at the current state within the Northeast LIN and perform a similar quantitative breakdown, we see that of the 497 potential NSCLC patients and those that would be ALK positive, which is 16, there is a shockingly low 12% capture rate, thus only detecting 1 ALK patient that would be prescribed Xalkori. So after speaking with healthcare professionals and technologists within Health Sciences North in Sudbury, Ontario, we were told that of the 497 potential NSCLC patients, annually only 60 are being screened for the ALK mutation, thereby giving us the shockingly low 12% screening rate. So our proposal is the implementation of a pilot study that would use medical education as a tool in order to increase ALK testing within this Northeastern LIN. Now, in order for our implementation to be successful, there were some assumptions that we had to take into account. And I'd like to direct your attention to the fourth assumption, which has been alluded to earlier, in that all ALK-positive patients that we would detect would be prescribed Xalkori. And this is a fair assumption to make, seeing as how Xalkori is currently the only FDA and Health Canada approved drug for this specific clinical indication. Next, we're assuming that the physician attendance rate for these medical education programs would be 30%. And lastly, we're assuming that the change in physician performance as a result of these medical education programs would be 70%. And this assumption has been made based on extensive literature review that that showed that over 160 different medical education tools elicited a 70% change in physician performance with regard to the medical education program that it was intended for. So let's revisit the current state of the Northeastern LIN with the 12% capture rate. Now, as a result of our medical education tool, we project that of the 16 ALK positive patients that are projected to exist, using the 70% capture rate that would occur as a result of the medical education tool, that we would be able to capture 10 additional ALK positive non-small cell lung cancer patients, thereby exhibiting the vast additional revenue that this poses for Pfizer in terms of Xalkori prescription. So, our medical education tool would be a continued medical education. Now, this can be referred to as an educational activity that is used to maintain, develop, or enhance the knowledge, skills, and professional performance for the physicians that it is intended for. Now, our CME pilot program must be accredited first for family physicians who actually require 250 CME credits to be obtained over a 5-year cycle. Similarly, specialists require 400 CME credits to be acquired over a 5-year cycle, thereby incentivizing the physician attendance rate of 30% that we hope to attain. Now, to dive further into our pilot study layout, we plan to sponsor 2 conferences, the first being the Regional Oncology Conference and the Better Breathing Conference, both of which would be located in Sudbury, Ontario. Now, these would include an 8-hour interactive and case-based workshop that would be accredited by the College for Family Physicians of Canada, as well as the Royal College for Surgeons and Physicians and the Northern Ontario School of Medicine. Now, their classification refers to 1 hour of academic learning to be equivalent to 1 CME credit. Therefore, our pilot program would provide both physicians as well as specialists with 8 CME credits by attending our program. And this program would consist of oncologists, pathologists, as well as Pfizer representatives. So just to get an overview of the timeline for our pilot study, in January, we would be pitching this proposal to Pfizer after obtaining CME accreditation. And then in February, we would be developing the CME program material as well as the promotional items. And then from March up until the conference date, this time would be allocated towards marketing and advertisement in order to attain that 30% physician attendance rate, which would be divided into 2 conferences. So this would mean that only 15% attendance rate would be required. Then in June and October would be the conferences themselves, after which there would be a post-conference analysis done using the 7 levels Moore's CME outcome measurement to determine how effective our CME was. So now let's look at a financial analysis of our pilot study program. Illustrated above is the income statement for the period ending 31st December 2016 that factors in both years in which our pilot study would be conducted. We project a revenue of $396,000, and this takes into account the sales that would be generated for the additional ALK positive NSCLC patients that we would detect as a result of our CME program. Now, this is a very conservative estimate because we've also taken into account the fact that Health Canada reimburses Xalkori only up until tumor progression. So the phase 3 clinical data has shown that the median time for progression for a tumor is 7 months. So we've only attributed a 7-month sales in our revenue. Now, we've also broken down the costs, which total approximately $203,000, thereby giving us a net profit of $193,000 approximately. We've also calculated a positive net present value of approximately $136,000 using the discount rate of 8%, which has been extrapolated from the industry average that is currently being used in the pharmaceutical industry. Now, this positive NPV is a conservative estimate as well, as we have factored in the 30% physician attendance rate as well as the 70% change in physician performance as a result of our CME program. We further conducted a break-even point analysis for our financial analysis. And if I can direct your attention to the y-axis, this basically shows the sales and costs on a yearly basis in thousands. And on the x-axis is the time of implementation for our pilot study program. Now, if I direct your attention to the costs of our pilot program, illustrated in red, we see a steady increase in the beginning of costs, and this can be attributed to the development of the CME program material, as well as the marketing and advertisement that would go into developing the CME program. After 2 years, we see a tapering off as the funding would decline after our pilot program. Now, if I direct our attention to the sales, we see that in the beginning, we have not attributed any sales to occur up until the end of the first conference that would occur midway through year 1. After that, we projected an increase in sales due to the additional ALK-positive patients that we would detect. However, this too levels off, as we have determined that after our 2-year program, ALK testing would become the standard of care as it is in the Toronto Central LHIN, and hence there would be not many additional ALK-positive patients detected. So just to wrap up, the solution that we have proposed is the implementation of a pilot study that would use continued medical education as a tool to increase ALK testing within the Northeastern LHIN. And I'd like to highlight the feasibility of our business proposal with a positive net present value and a reasonable 1-year break-even point. So what we plan to do is to address a critical gap that exists currently within the Northeastern LHIN, and that is the lack of widespread adoption for ALK testing. Now, what does this mean for patients like Doug? By increasing the frequency of ALK testing, we'll be able to detect more patients like Doug who possess an ALK-positive non-small cell lung cancer. And as a result of detecting him, he would be eligible now for a more clinically effective, targeted, and personalized anti-cancer therapeutic, which at present time is the drug Xalkori. We'd like to thank you for listening to our presentation, and we now open the floor to any questions that you may have.
Hello? Oh, you can all hear me. Okay. All right, so I have a few questions actually that I'm going to kind of wrap up into one. If you go back to your financial slide, I'm going to start with the hardest part first. I guess one question just to start is, have you spoken with an accreditation body on the cost of putting together an accredited program?
We've actually researched the previous programs and the funding for them, as well as the actual sponsorship levels. We researched it by looking and contacting Health Sciences North on previous conferences they had held, and what the gold sponsorship level attained them, as well as the outcome measures that they usually use to measure the actual physician rate.
Okay, and sorry, where does that show up here? That actual, I guess, payment or whatever that you have to make in order to have an accredited program go through?
Under our gold sponsorship, that's where it would come in.
Okay. Now, the other piece to this is that you cannot actually build the accredited program yourself. And in addition to that, you need to perform a needs assessment survey beforehand. So essentially, if a need is not demonstrated by the physicians there, if they don't agree that they need this, then it doesn't go through. So have you spoken with any of the physicians there to determine that if in fact this were to occur and a needs assessment were to be produced, that they would vote basically yes, that they would need this?
So currently, actually, within Northern Ontario School of Medicine, they were accredited 5 years in 2012 for the next 5 years for implementing CME pilot programs. And After speaking with both healthcare professionals and technologists at Health Sciences North in Sudbury, which is basically like the hub for diagnostic testing, they said that they do have the resources to conduct ALK testing because while it's a larger population, Toronto Central has a lot more patients coming in from different places. So resources is not the main issue. The main issue is the fact that the physicians, so for a patient like Doug, when he first goes in, it's the referral for biopsies that's lacking. And we found that the best way to intervene or get the best outcome would be conducting a CME, because then we would have, and this is why we factored in not only accrediting it just based on specialists, but that's why we also included family physicians, because they're the people that are going to have the first contact with the patient, and they have to refer him for a biopsy sample for additional molecular testing.
Right, but if they were to receive a needs assessment survey that basically goes through, because you can't be, that's the one issue with doing an accredited program, right, is you can't say, do you need this specifically? You have to say, here are the different areas, in which of these areas do you have an educational need, and then from there can you specify what you're looking for, and then hopefully what you're looking to do falls into one of those categories.
Yeah.
So I suppose my question is really, did you get confirmation from any of those healthcare providers that if they were to receive a survey like this, that would be an answer that they would put down?
So talking to the director of the medical and cytogenetics lab there, they did address the fact that, yes, there is a need there for increasing biopsy samples, and that was specifically the restriction that they said. And they said talking to other physicians, they also noted that, yes, this is the actual kind of bottleneck in the issue, and that's where the need was addressed. So they did address the need. I wasn't able to contact any further oncologists on that issue, but the medical director and the ones that she had referred me to did address that issue and illustrate the need for it.
Okay.
Hi guys, great presentation. So I want to build off a point you just mentioned. So you noted that referrals for biopsy are the issue, and if we go according to your plan and you increase referrals and testing, do you think the North has the resources necessary to handle all those tests?
Yes. When I was talking to, like I said, the medical director for the two labs that are there, they said they do have the resources available because currently they're also doing biomarker testing for HER2 as well. So
yes,
it's not
to
just for
increase, not for the current status, to increase to the levels that you need. And do you
Yeah,
think
they would be able to. They said that the only issue they have is the biopsy samples. They have the facilities and they have the technologists. And talking to other hospitals as well, they also said that this didn't increase the cost the lab incurred because all the costs for the tests are reimbursed. So they have the facilities and they're also reimbursed, so it's at no cost to the hospital itself.
And is the reimbursement volume-based? Would the government, raise a red flag in terms of you increasing the testing for
Yeah,
that?
exactly. So the great thing about the ALK testing is that it uses the same diagnostic techniques that are used for HER2, for example, and that's really widespread within all of Canada, right? So basically you'd be using the exact same diagnostic assays, just changing the antibodies, which are actually really cheap in terms of ALK. So it wouldn't be a matter of, even if you were to increase the volume, they already have in place a lot of different, they have the resources available to target the biomarkers because you're using it for HER2, for example, EGFR. So, we would be detecting, as we mentioned, maybe 10 patients on an annual basis, so that wouldn't burden them.
Okay, fair enough. My other question for you is regarding competitors. So are there other drugs coming to market in the near future, and do you think you'll be helping them by doing this? How will you mitigate against that?
Just go to the slide. 1 second, we're just going to get to the slide.
Just exit out of the presenter mode.
Yeah, and just go down.
Which one is it? Can you find it?
Oh, yeah, sorry.
Sorry, there, found it.
Found it. So we have Zycadia, which is by Novartis, and this is currently available in only only US, sorry. It's only FDA approved, and it's actually only approved for those that are resistant to Xalkori. So it actually is not a direct competitor in those terms, and it's actually not even reimbursed in Canada because they don't consider it cost effective. Furthermore, currently there are other molecules that are competitors for Xalkori, but they're still at the initial stages of the clinical trials. So there's some in preclinical, there's a few in phase 1. There's none that are present in phase 3 currently.
Okay, thank you.
I'm just going to bring up our other welcome slide.
Hey guys, good job. It's a tough topic to work with. Going back to Annie's point about the laboratories in the northeastern Lynn, from my understanding, Cancer Care Ontario has to do an assessment of the laboratory in order to grant it accreditation to run the ALK1 probe. Do any of the hospitals that are in that Lynn currently have that accreditation, and do you know how long it would take to get it, if not?
Well, Sunnybrook Health Sciences North does have that accreditation for that specific Lynn, and they do other biomarker testings.
So Sunnybrook?
I know Health Sciences North in Sudbury, Ontario.
So they're already accredited to do ALK?
Yes.
And are they the only one in that region?
There's another one, the medical school there.
I think it's the Northern Ontario Medical School.
Yeah.
Okay. Do you know approximately how large the northeastern Lynn is? And do you think that 2 hospitals would be sufficient to cover?
What is that in terms of like kilometerage?
Yeah, well, I mean, I don't need an actual figure.
I don't have that number. To be
No,
honest.
I don't expect you to. I'm just trying to bring light the fact that the population density in that part of the province is very low. So do you think that there's an issue with the proximity of these reference laboratories?
So currently, Health Sciences North is the only specific diagnostic program for molecular testing. However, there are other diagnostic programs within that Lynn that are more geared towards radiology, and they're actually kind of speckled throughout the Lynn. Now, when they get immediate chemotherapy, they do molecular testing, but they take the biopsy sample and they send it to Sudbury on a mean of transportation costs, and then it's tested there, and then the results are actually telecommunicated back using the Ontario telecommunication network from hospitals.
Cool. One more question. Can you go back to the slide where you're comparing northeastern Lynn to the Toronto, and coming up, you derive your adoption value, or how many patients in need are actually receiving the medication?
Yeah. This one? Okie doke. So, yeah. This slide?
Yeah, perfect. So what did you use to determine the 100% capture rate and 12% capture rate? Is that just canvassing opinions of physicians? Or?
Yeah, this was the opinion of our extensive research, talking to not only doctors in Toronto as well, but also other ones in Mississauga, such as Dr. Higgins and Dr. Legal, who are both lung cancer specialists and the head of their oncology departments, who said that as soon as a patient is tested positive for non-small cell lung cancer, they immediately get reflex testing done, and then as soon as they are ALK positive, they're immediately put on Xalkori.
Okay. Now, can you go to the graph where you show the trajectory of patients that have undergone the FISH analysis, and then how many people have been prescribed Xalkori?
It'll show up.
Yeah, perfect. So is your data, see, this shows from May to essentially a year and a bit, how they've risen exponentially. Is your data based on, does it take into account, if you were to extrapolate this line into the future to the point where you start to see the benefits of your program, how you may no longer be dealing with a 12% rate of success, how it may be much larger by that point because of the improvement we're seeing over time? Or is your data using this snapshot?
Well, the 12% data is from our current conversation with the Health Sciences North in Sudbury. So that's the current figure. Whereas this one is from May 2012 to June 2013. So in terms of like what would happen if we extrapolated this data, because I don't have the data after June 2013, I would not be able to speculate on that.
Right. But it's safe to say that you may be at risk of overestimating your values, because by the time the effects of your program take place, you're already going to see a higher rate of ALK probing. And
I mean, our starting point, again, was the 12% capture rate, as per our conversation with the people at Health Sciences North, and that's our starting point.
So that represents right now.
Exactly.
So, and you said it would be about a year until you started to get your accreditation program running?
Well, we're going to start in January, which would be like four months from now. So about 2 months from now.
4 months from now.
2
Oh,
months from
okay.
now, yeah.
And then the entire program lasts how long?
It's 2 years.
2 years. So
Yeah.
in 2 years, the data that you're working with right now probably won't be the same. Like you have a natural progression towards improvement.
Okay.
So,
I understand
You
your
see
point.
what I'm saying?
Yeah, I do understand your point. So our pilot program is 2 years. However, it's split up into like on a yearly basis, right? And we have taken into account the fact that, and that's why you saw the revenue kind of level off. And it's because we realized that. So we're assuming that after our 2 years, which is exactly what you said, after 2 years, we're assuming that it's going to become the standard of care as it is in the Toronto Central LHIN. So as a result, we've calculated our revenue based on the additional ALK positive patients that we detect. And that's why it kind of leveled off, right?
that's why you plateau. Okay.
Yeah.
And just as a last point, could I see your sensitivity analysis? Please.
Well, let's just show them this. Yeah, throw it up there. So this is our sensitivity analysis, and we just compared it along 5% ALK prevalence and then 3% ALK prevalence. And since we took the most conservative estimate for every other variable within it, such as the attendance rate and progression rate, the only ones that we had to worry about were these two.
Okay, thanks.
Hey guys, great presentation. Back on the slide when you were comparing the Ontario LHIN and the Northeastern LHIN, there was something, you were comparing accessibility where you said the Northeastern LHIN had low accessibility. Can you define the exact criteria for what accessibility it means?
I can't really describe it based on pure range to hospitals, so I wouldn't be able to give you an exact value on how we got to our accessibility.
I mean, like basic, can you explain what you mean exactly by accessibility?
It was derived from population density.
So from my understanding, you're saying that for a certain population, the percentage of them are able to access this testing will be low compared to Toronto. Is my understanding of that correct?
Could you just reiterate that?
So what I'm saying is that percentage of population that have accessibility to the FISH test is low compared to that of Toronto Central. Is that a correct understanding?
Yes.
Okay. So based off on that, what exactly was the original factors that led to Toronto having such high accessibility compared to Northeastern LHIN?
They have, like I highlighted in the table, there's 5 diagnostic programs there. And with a high population density, they don't have to travel far. So they have a lot of accessibility to these resources. And this was based on, like I said, the diagnostic programs, but as well as the incidence rate for the ALK mutations, showing that they can easily get to these areas.
And just one more thing, if you notice, 4 out of the 5 are within UHN. So that's a very close proximity in which biopsy samples could be transferred.
And this will be basically within, it's pretty much just, so in a geographic area, Toronto Central, LHIN, roughly how big is it?
Oh, wait, it's back here.
All right, thanks.
So it's just the little guy, number 7 there. That's the Toronto Central one.
Okay, now the context is there. And this kind of goes back to Dave's point earlier about the difference in terms of geography. And I guess my final question would be that, so why exactly hasn't the knowledge of these techniques, because that's my fundamental understanding is that it's the knowledge that is missing in Northeastern compared to the Toronto area. Is it just because in Toronto the doctors are more likely to talk to each other because of close proximity? Is it just because there's a higher number of specialists, higher number of key opinion leaders in that specific region compared to Northeastern Lynn?
Well, I would say yes, that's one of the reasons. And I mean, physicians, when they come out of medical school, because of the new developments in the field, they would need to be constantly updated about the new developments and implement those developments in their practices. So distance, I would say, is one issue. And I mean, physicians in Toronto probably are exposed to more conferences, to more workshops. So that would be one reason why doctors in Toronto region are more aware in terms of referring these patients for different testings.
And has Pfizer run the kind of workshop you guys have talked about before in that specific region?
I'm sorry, say it again.
Has Pfizer run the type of programs you guys are talking about in the Toronto region?
Not to our knowledge, no.
Not to our knowledge, okay. Okay.
Hey guys, good presentation. Just have a couple questions. The first one's regarding, so you guys mentioned that the big issue is that the physicians, first-line GPs or oncologists, they're not sending enough people for a biopsy, right? And you're basically here by doing like a CME, or you're saying that they don't know that they should be sending it to a biopsy. Like, are there any other reasons? Like, is it a hassle for the physician? Like, would there be any other reasons why they wouldn't be sending the biopsy right away?
Currently, we aren't aware of any other reasons. From our knowledge, it was just the fact that usually when they progress on chemotherapy, they are either transferred to a different chemotherapy line that can often be platinum-based again. And so usually they keep progressing, and then after that, they just do palliative care from our knowledge. So the issue is only in education and referring biopsy samples, and that was the only issue that we found.
Right, so I guess.
And one of the reasons is that in one of our conversations with one of the pathologists here in Toronto, he mentioned that because of the low rate of ALK mutation, some physicians don't refer patients to oncologists for further testing. So there's that gap there. And we believe that by CME, we can close that gap. Okay,
that's a good answer. So the second question I had was, so if you're running your CME, I guess what's the plan to reach to physicians, and why would they go to your accredited program as opposed to other accredited programs they could go to? And also, because of the geographic region, is it easily accessible by the physicians to go to these conferences? Like, what if they have to fly in?
So when we're looking at accessibility for these physicians, there's actually already a network that allows communication between hospitals. So if we were able to advertise, say, at the Northern University Medical School, as well as Health Sciences North in Sudbury, which is what our marketing costs were attributed to, they can communicate this between hospitals using the OTN of the Ontario Telemedicine Network.
Right, but to attend the actual conference, is it going to be like an actual conference or like an online
it would be an actual
learning
conference.
On demand? Okay.
So we also were considering a webinar because currently that sometimes is deemed to be more accessible.
Right.
To your point, right? However, we wouldn't be conducting a conference on our own. Our CME pilot program would be incorporated within these conferences, which are already ongoing. Like, they already occur. So we'd kind of just be going on with that. So we wouldn't be advertising to everyone to come to the conference. They already have like a base, we assume, that would come. So, and as we mentioned, right, it's a 30% physician attendance rate, but that's divided. We've mitigated that risk by by being part of two conferences. So that would be a 15% attendance rate for either one. And in our sensitivity analysis, you saw that it would only actually require 8% for us to break even.
Right. And just curious, one more question. You guys have any idea what kind of speakers you'd be inviting to do this kind of talk?
Usually like a key opinion leader, such as a clinician or an oncologist in the Toronto Central area who is already actively doing this reflex testing and can attest to its efficacy for detecting ALK-positive patients.
Okay, thanks.
We're going to close it there, guys. But great question. Now we're going to open it up to the floor.
Good talk, guys. So thank you very much for your talk, and I apologize for having quite a few questions. I'm just quite interested in this area myself. I guess just a quick side note, I was a little confused during your presentation whether the stance of your team is from a commercial perspective or a medical perspective, because that would be very important in the context of sales, and I'm going to assume it's commercial because you use the word sales, in which case I would advise you probably not to talk about quality of life in one of the slides I believe Alex presented, because that's actually not mentioned in your monograph, so you'd be doing off-label marketing as a result of that. But let's move on to something that really struck out to me. It was the slide about your prices, the one with the 160 grand and everything else. If we could go to that one, please.
Oh no, going the wrong way. This one?
Yep, thank you. So I just had an issue with your, I guess, total cost number there. Because one thing I noticed, which was quite interesting for me since I do marketing, was that your advertising materials cost was $1,900. And I was wondering where you got that from.
So we estimated our advertising cost as making the infographics for the promotional material that we could include for our delegates, as well as the actual booth setup and the posters that would be using at the conference itself.
Okay, so I'm going to let you know now that, so that was a booth and a sort of like, I guess, infographic pamphlet you're going to hand out to physicians. Just with the pamphlet, you're looking at about 30 grand there. So you're quite off your estimate. And then there's the booth, which will probably be about 20 grand or so. And you will probably be limited to about 2 sales reps at this conference, which makes me doubt the ability of your sales reps to contact all of your customers there. The other price I had an issue with was with the speaker honorarium, which he or she, depending on how many doctors you have, was only paid $4,000. And to my knowledge, this price would, let's just say it would be substantially higher, again, throwing off your budget costs by quite a lot. Do you have any responses to that?
So when we researched speaker honorariums, we were kind of going on current trends in that area, and we researched it between usually about $2,000 between $5,000.
Per hour or per talk?
Per talk, and we'd only be doing one talk with our speaker.
And so I know Evan and Jason next to me have a couple questions, so I'm going to just ask one more for now, and maybe if there's time I'll come back. But have you talked to any sales reps in the field that sell non-small cell lung cancer products? And what have they told you about changing physician performance percentages by 70% with just 2 CME programs?
Well, we talked to the commercial lead of lung cancer at Pfizer Canada, and he pretty much agreed with the problem that we posed and that there are these barriers. And we did not specifically discuss the CME program itself with him. But yeah, so to answer your question, no.
Just to further address your question, as you said, the booth would cost about $30,000, but when we looked at sponsoring this event, that was actually included in the cost of the gold sponsorship for the event.
So the sponsorship gets you a booth spot at the event, but it doesn't promise not only the creative design of your materials, the agency fees that you'd pay to an agency such as Ogilvy or CPC Healthcare, and it also doesn't cover any costs of transporting these materials from your home base, which would be, I believe, in Montreal or somewhere in Quebec, to Sudbury with another agency such as Reveal Marketing.
All right, thanks, guys. Good talk. So can we flip to the slide where you guys have your list of assumptions, please? Okay, so assumption number 4 there assumes that all ALK-positive NSCLC patients will be prescribed Xalkori. Okay, so how are you certain that all ALK-positive patients meet the indication on the drug? Yeah.
We based that assumption based on this graph here. So If you look at the yellow, the green and light blue lines at the bottom of the graph, as you can see, they overlap. So the green is the number of patients that were ALK positive, and the light blue is the number of patients who were prescribed Xalkori. So pretty much all ALK positive patients were prescribed Xalkori. So we based that assumption based on this graph in this particular study.
Follow up to that as well. Have you also considered the possibility that your patients might also be captured by clinical trials for other drugs as well?
No.
We haven't, but oh, do you mean like other drugs specifically for this indication?
Sure, or for these sort of patients, non-small cell lung
Well,
carcinoma.
currently, Xalkori is the only drug approved in this indication for ALK positive non-small cell lung cancer. So, no.
Can you guys go back to the slide with your assumptions on it? And I wanted to talk about that and the slide after that. And how you got to the 70% increase of adoption number. I may just be missing the math here. But you said a physician attendance rate of 30% and a change in physicians of those that are attending, I'm assuming, of 70, which is a change in rate of 0.21, which is just 21%, not the 70 that you have on the next slide. So am I misunderstanding your assumption 6 there?
So that rate was assumed after it becomes, this event had been held multiple times. So analyzing the entirety of the program, not just the initial year, but over like a 5-year period.
Okay, so that, however, is that captured in your cost projections? Like that's, so what you've done for the 2 years then, did you use this 70% capture rate or did you use a lower
When
one?
we calculated our revenue, we included the 30% attendance rate as well as the 70% capture rate. So we did
you
take
used
that into
21%.
analysis, yes.
Okay. My second question, is that okay, Parker? Sorry, Dr. Parker. So how many oncologists or physicians are actually seeing these patients in Northern Ontario? Like an actual number or order of magnitude at least?
So from my communication with the Health Sciences North, they only do 2 or 3 confirmatory FISH tests a year, which means on that number, they're usually only seeing about 2 to 3, and that's based on the capture rate that we found, which was only 12.1%. So that's where we extrapolated our data.
So how many physicians is that? Because if you're assuming 30%, you're kind of basing that on a large numbers assumption. If you've only got 5 physicians who are seeing patients and 4 of them happen to decide they don't do CME or these CME,
so specifically at Health Sciences North, I believe there's 299 physicians.
Oncologists or physicians?
They're all included, and I believe there's 2 medical oncologists and then 2 surgical oncologists.
So 4 physicians are seeing lung cancer patients in the northeastern LHIN?
Not only just, those would be the oncologists, but there's also the family physicians, as we pointed out earlier, who are the main target to increase biopsy sample reference.
Okay, are they prescribing Xalkori?
No, they aren't. They would be referring the patients. For example, so LHIN is a huge area, and the designated hospital is in Sudbury. So physicians in other parts of this LHIN would refer the patients who live in the rural areas of LHIN, would refer their, basically these physicians would refer these patients' samples to the designated hospital, which is Health Sciences North, for testing. So basically, we would like to have those primary care physicians located in all over the land to basically send the samples of patients to the designated hospital.
Okay, so it's those physicians that are prescribing first-line chemotherapy. It's the family physicians outside?
Well, eventually, yes, after the diagnosis or confirmation.
So no matter what, they are sending a sample to be looked at by an oncologist in Sudbury. So really,
yeah,
Sudbury
they
is where
will
we need to
still
target.
send the samples to Sudbury for testing because they don't have the resources there.
Okay, so I guess my question then is why are we targeting this diffuse field of physicians when there's a bottleneck in Sudbury that's doing the testing that can be potentially?
Well, there are resources in Sudbury, but it's just that they don't receive enough specimens. So if we increase education among the physicians all over the land, then more patient specimens would be sent to this hospital from all parts of the land.
How does it work to figure out what's going to be sent? Like, Sudbury can't just say when you're sending us a sample, make sure you're sending this. This is now what we test when we test samples, or?
Can you repeat the question, please?
Sorry, I know that was a little all over the place. So when the family physician sends a sample to Sudbury, Sudbury likely has some sort of list of, we expect to receive this, this, and this from you. It needs to be in paraffin. We need this many slides, that sort of thing. I would assume you guys can tell me if I'm wrong there. And if that's the case, why can't the Sudbury facility just change their specification so that they now do the ALK testing?
You want to answer that?
Yeah. So based on Cancer Care Ontario's lung cancer tissue pathway that we adapted for our treatment algorithm, so as I mentioned, a biopsy would be taken and that would be subjected to histopathological analysis, and that's when chemotherapy starts once they determine that they have lung cancer, right? So the bottleneck is what happens there, in that after they put them on chemotherapy, at least to our knowledge, within this northeastern limb, usually that's just continued until the tumor would progress, after which case they'd be put on a second-line platinum-based chemotherapy. So what our CME program is hoping to implement is that they won't just stop at that first-line chemotherapy and then allow it to progress and then just give second-line chemo, because that's not very effective. Rather, they would obtain another biopsy sample that they would subject to molecular testing at Health Sciences North to determine that they would now be eligible for Xalkori, which we showed was a lot more clinically effective than just doing a pemetrexed or docetaxel.
Okay, so that, I believe, answers my question. Is there a difference between the content of what's in the first biopsy and what's in the second biopsy? It's a different type of biopsy?
Yes.
Okay, thank you.
Hey guys, I got two questions. Hopefully they're quick answers. The first question is regarding the two conferences that you chose. Are those currently like CME conferences, or are those conferences where you go chat up with all your friends and hang out?
They're currently accredited CME conferences already.
Okay, so how many CME other workshops happen at the same time then? For those conferences, right? So those conferences are happening, and I'm guessing that there's maybe 5 or 6 different CME courses going on?
At the time?
Yeah, not many.
In the Sudbury region, there's not that many conferences that are held. There's only a few that are progress, and at that point, to my knowledge, there's no other CMEs that are held during that period of time.
No. So I'm communicating my question. So what I'm asking is, are there other courses happening at the same time as that conference?
Where?
Or is the conference itself like the CME?
No. So the conference itself is not solely our CME program, right? So that's why we've attributed the fact that we want that 15% at least physician attendance rate. So we're not sure about how many would occur, but to my knowledge, it wouldn't be more than about 5 or 6, because it is like an 8-hour based interactive and case-based workshop, right? So basically what it would incorporate would be including, for example, physicians as well as oncologists, and have them go through, case-based analyses. So if someone was to present with this, would you refer their biopsy sample? Okay, now when you did, you'd conduct these diagnostic assays. How would you determine the split versus non-split? Just to kind of increase their awareness about the ALK diagnostic testing algorithm, just so that, that would increase awareness and thereby increase ALK testing and then diagnosis and then Xalkori prescription as a result.
Okay, so we'll just move on. My second question is about, did you guys contact any physicians in the Northeast Lynn area? So you mentioned that you contacted the people who do the fish testing, and that they did X amount of tests, and it wasn't a lot. So clearly, there was some sort of problem with the prescription of getting a biopsy. So did you talk to physicians saying do you actively try and get your patients to get biopsies, or people who have these problems get biopsies? Because perhaps it might be maybe a patient compliance problem where someone says, "Oh, Darren, you've got lung cancer. You should go on chemo, and then also we'd like you to get a biopsy." And I'd go, if I'm not a scientist, I'd be like, "Well, I'm already on chemo. Why do I need to get my lung cut open and like get a biopsy?" So maybe like what I'm saying is like, is it that you've made the assumption that physicians are not prescribing the biopsy, where it might be that patients are getting the choice of doing the biopsy, and they're saying, "Well, screw that, I'm already on chemo."
So you're saying basically the patient would refuse?
Yeah.
But that would also be the case in the tronolene, which is our control. So that will be taken care of. Because that same thing can also happen with patients who are in Toronto, and we take tronolene as a
that might
reference.
also because the accessibility issue, right? Do I want to go drive 2 hours to the nearest hospital, or to the only hospital that will
their
cut me open?
specimens would be sent, would be shipped for testing at the designated hospital, which is Health Sciences North.
Okay, so back to my original question is, did you guys go talk to physicians about their, I guess, their personal algorithm of treating their patients?
From talking to one of the oncologists at the center, they didn't talk about the amount of referrals they did, but they said that usually they progress on chemotherapy and just keep progressing on other chemotherapies or general chemotherapies from my conversation with them. And they said that at the moment, it's not really patient resistance. And the ones that are at Health Sciences North are more aware of the algorithm. It was trying to target areas just outside of that as well.
Okay, thanks.
Hi guys, good presentation. I kind of have one question and one comment. I'm kind of confused why you chose Ontario, considering they probably had the most restricted in terms of their funding for Xalkori across Canada, first of all. So it probably would have been better, I would think, to pick a province like BC or somewhere like that. So you can address that if you want, but that's just a comment. My second question is, in Ontario, they have a time-limited funding. And can you just clarify, I think you said that you're assuming that they're going to fund it for 7.5 months, right? Or 7 months or something like that?
So under our research with P-Coder, it's reimbursed as a second-line therapy up until they progress. And that's where we took the 7.7 months of clinical or the median progression-free survival for these patients into account.
So P-Coder only gives a recommendation, and then the provinces will decide
I'm aware of that, yeah.
how it's funded.
Yeah.
But in Ontario, it's time limited. So as of October 1st of last year, it's up to the executive officer to decide how long it'll be funded for. So up until 1 year is the maximum right now, and then they go for a renewal. So I'm just wondering why you picked the 7 months in particular. Because was it off the clinical trials? Like, what was that 7 months off of?
So basically, right now, Health Canada only reimburses Xalkori as a second-line treatment up until tumor progression. So after those 7 months, it's no longer reimbursed, and that's because they're going to progress. Their tumor would progress, so they'd be put on another third-line chemotherapy. So I'm not sure if that answers your
question. No, I understand that. That's not what I'm asking. Okay. I'm saying that right now, in Ontario, it's up to the executive officer how long it'll be reimbursed for. 1 year is the maximum, and it's up to them. It's just, did you talk to physicians to actually find out how long it is actually being reimbursed for their patients?
No, we did not, and we did not ask them that, and that's why in our revenue and stuff like that, we only incorporated it for 7 months.
And because my concern is that 7 months is a bit high, and I would think that you should find that out because it could be lower, and that should have been worked in probably to your sensitivity analyses.
I just have one quick question. It's very simple. So you propose that you can do a CMA, 2 CMEs to increase adoption by 70%. Do you have any evidence or examples in the industry at large where doing 2 CMEs have increased adoption by even 50%? Or even 1 CME?
No, but just based on our research, that's where we got that number from. We didn't find any real-world examples.
Okay.
Questions here? Okay. pass it down, please.
Hey guys, great talk. I thought you did a great job. I have a quick question. I don't know. It's just for my own clarification, actually. So when you were describing the FISH assay, you got really into detail about like the DNA genomic side of it. I'm just curious to know, like at a higher level, how do you tell which cells are normal and which are cancerous or tumorous?
Are you asking how we differentiate the cells,
Yes.
whether they are tumor or normal?
Yeah.
By looking
just
at the image?
Based on that, yeah.
Okay, so when you do FISH, you get the sample from formalin-fixed paraffin-embedded tissue. And so basically, you would cut a tissue section from a paraffin block, and you would do your FISH assay on that section. Now, you would also cut another section from that paraffin block and stain that section with hematoxylin and eosin staining, which would be H&E staining. And when you stain the tissue with H&E, you can, when you look at it under the microscope, you can say which cells are tumor cells, which cells are stromal cells or normal cells. So then what you do, because you cut the FISH section and the H&E section from the same paraffin block, you can compare the two. For example, if you see 5 cells in your H&E section, if you say, okay, the 2 cells at the top are tumor cells, then you're going to look at those 2 cells at the top in your FISH image. And that way you can say, okay, these 2 cells on the FISH are the cancer cells. So you're going to focus on those 2 cells specifically. And you can see whether there is a rearrangement or no rearrangement in those 2 tumor cells. And it's possible that one can have rearrangement, one will not have a rearrangement. And I guess there could be another question there, like how do you then, where is the cutoff? Like how do you determine whether this patient should be diagnosed as ALK positive or not? And the cutoff for that is 15%. So if you're going to look at 100 tumor cells on your FISH, then for that patient to be diagnosed ALK mutant, you need to have minimum 15% of those tumor cells. 15%, 1-5, to have ALK rearrangement. So if you have minimum 15% of cells that have ALK rearrangement, then that patient is diagnosed as ALK mutant.
Okay, thank you.